So, you worked on the basic mineral nutrition, like with boron and others, but you also worked with potassium and many other nutrients,
Oh yeah,
phosphorus.
actually, when I started my PhD, uh, I got a couple of papers, one from Israel and one from the Netherlands, and the guy from the Netherlands wrote like 100 pages,
Oh, wow.
and then the group from Israel wrote about 3 pages, but they had a graph, they had a drawing in there of the hypothesis
Mm-hmm.
of what plants send up the xylem and then what plants send down the phloem.
Mm-hmm.
And I noticed that potassium was going both ways, and it's a single plus charge. It's a small element, and it doesn't stick anywhere, but it's a positive charge, and it's really important in plants, going up the xylem from the roots to the shoots, and then back down again with the sugars and organic acids in the phloem, and it kind of makes a cycle in plants. So I learned that it's really important to build a model or a diagram which has all your thoughts in it. And so that's what really directed my PhD program when I got my PhD, my research. And then I've kind of I like drawing models and trying to figure things out that way. And
Yeah, they all interact together.
yeah, and then you can design experiments to test that.
Right. And then you find that high protein crops need a lot of potassium because of that.
Oh yeah. I have a chapter in a book, Potassium in Agriculture, and it's about potassium and protein, and yeah, you can look at dicots like soybean, and they're very high potassium, high protein, and then you can do grasses separately. They have less protein, less potassium requirement. You can it really goes together.
Yeah.
And the reason is there's 20 amino acids in most protein, 20 different ones, and 2 of them have negative charges. And so they need to be neutralized by potassium. So they have 2 potassium sticking on there. So you can actually do some calculations, which I've done, and I did in that chapter, and it all comes out in the wash, plus and minus.
Wow.
But even at Oregon State in biochemistry, we had to go there, and because I was working on potassium activation of enzymes, and they had a whole room designed for finding out where potassium was, because as a soluble little plus charge, it's so hard to find out where it is,
Yeah.
So it's very difficult.
Hi, Del Blevins in his interview stated that high protein crops need more potassium, and it's related to nitrate transport. So here we have a corn plant and we have nitrate and potassium out into the soil solution. Well, they have no problem getting into the root system. The issue is, how does the nitrate get from the roots up into the upper portions of the shoot of the plant? Well, potassium and nitrate can travel up the xylem together, and when they do travel and get to the destination, the nitrate disassociates itself with the potassium, and now it's free to be converted into amino acids, into proteins in the leaves. These can be stored in the seeds, or they can be in the plant, giving lots of protein. Now, the potassium then is free. Well, what happens though is that photosynthesis is happening. That means CO2 is produced into compounds of carbon that the plant can use. In this case, we have malate, which is negative. The potassium and the malate join together and travel down into the plant, into the root system. Now, the root system is being fed a carbon source, so it can be used for growing, and then the potassium then is then recycled back to bring more nitrate up to the plant, and then once it gets there, it's used to bring more carbon compounds back down to the root system. So the root system is happy, it's growing, the proteins are being developed in the upper part of the plant. Also, what happens is this carbon compound doesn't necessarily always stay in the root. It can be exudated, meaning it can go from the root out into the soil solution. Now, why would it do that? This feeds the soil microorganisms. And many of the soil microorganisms are very important for the transformation or the conversions of nitrogen, sulfur, and phosphorus into forms that the plant can use. Also, many of these microorganisms manufacture compounds that stimulate the plant growth. So, to get high protein crops, like if you wanted a high protein grass hay for tall fescue or alfalfa, or if you want a high protein grain crop, you want to make sure you have plenty of potassium so it can transport the end source up the plant and also bring down carbohydrates to help the root system grow and also feed the soil microorganisms.
And then talk about the different nutrients. You mentioned potassium, and you found out in amino acids that some require sulfur, don't they?
Oh, yeah, that's another story.
Yeah.
Okay. So there's like 20 amino acids. I keep saying that. But 2 of them have sulfur, methionine. And and it turns out that soybean is low in methionine.
Is cysteine the other one?
Yeah, cysteine. And then grasses are low in cysteine. So it kind of works out. That's why you have beans and cornbread and beans make a good diet, because you're mixing the 2 types of proteins together.
Mm-hmm.
One's high and methionine, one's high in cysteine. But anyway, so a long time ago, I had friends at DuPont, and they were trying to beef up the sulfur amino acids in soybeans. So they sprayed them with every kind of sulfur compound you can think of. It didn't work. So that's when I got the idea, okay, let's shortcut it by using IVs. So that's when Larry Graybow was here, and his wife was a nurse, and he put the soybean intensive care unit at Bradford Farm it's one of the famous pictures.
Yeah.
Made all the agricultural magazines, soybean intensive care unit. And we had all these pediatric IV kits, and then we were lucky because down at the base of the stem, it's pithy, and there was an open spot where the soybeans would have a chance to drip. So we IV'd in methionine, made a huge difference in the soybeans seeds that were developing.
To be high in methionine.
High in methionine, yeah. The soybeans and what Larry showed by electrophoresis is that every seed has just a certain amount, 4 to 6 or something, of main storage proteins in the seed, and soybean had one that had no methionine, no sulfur amino acids in it. And so what Larry showed is that when we gave the methionine by IV, it quit making that one and made all the others that have methionine in it.
Really?
So soybeans are pretty smart if they have a source of methionine.
So it changed it.
Yeah.
Wow. And not only and that led to IV and boron later on.
Larry
shown.
Yeah, and then cytokinin. You did that.
Oh yeah, and we put calcium. We put lots of things in the soy.
Yeah.
Joyce always said, if I didn't know better, I think you're a drug addict because you always had needles in your pockets.
Yeah. Now, can I tell you a bad story about that?
Oh, okay.
So once I went to a meeting, and there was a young guy from another state, faculty member, and he more or less called me a liar because he had tried IVs on corn, and it didn't work. And this is in a public meeting,
Mm-hmm.
so I was not very happy with that guy. But then my friend John Boyer, who's a National Academy
Yeah.
of Science guy, he was at Texas A&M and endowed chair there. So he came up to see me in Curtis Hall, not Curtis Hall, in the ag building here. And he sat in my lab and we talked, and he says, I want to use those IVs on corn. And so he interviewed me. I told him all about how to do it. And he went back, and they discovered that corn is solid down there at the low stem. It doesn't have a pithy area. So he had his master's student use a little drill and make a little hole in the corn and make an area for a drip to happen. So they put a little rubber septum in that hole, put the IVs in, and it worked with corn.
Yeah.
But he was a National Academy member, and the other guy was just a young professor who knew a lot, But so he couldn't do it. He didn't figure that out, like John's.
Well, and that's a lot of people, it doesn't work, you give up. Well,
Yeah,
maybe
yeah.
your methods weren't right.
Exactly.
And we did some of that with cytokinins with Uncle Roy.
Yeah.
Roy Morris.
Yeah.
Got some nice publications out of that.
Yeah.
So, but we've had a hypothesis before. You do the experiment, it works. At least the data supports it. So you repeat it. That was always important.
Yeah.
To repeat it. And you find out later on that your hypothesis was wrong.
Exactly.
Because as you get better doing your experiment, you find out that it goes a different direction.
Yeah.
Often, that's even a better idea.
Oh, yeah.
Because the model fits better.
Exactly.
So you got to come in with an open mind.
Yeah. And it's amazing how that's hard sometimes to have an open mind and to admit that you were wrong the first time.
Yeah. Yeah, but
but that's science.
it is. It's science.
Yeah.
I think early on in your career, you didn't even bother with sulfur studies because we had so much sulfur in the atmosphere. You remember we were doing stuff with acid rain and
yeah.
and then we cleaned it up, which is great.
Well, there's a wonderful. Can I tell a story about that?
Yeah.
So we have a very famous guy in forestry named Rich Guyette. Did you know Rich?
I did.
Yeah. Well, I was on his committee. He's a local guy, and I think he worked with Harold Eugene Garrett. But anyway, I was on his committee, and then we would go on float trips every year. I knew him pretty well. But he was a forestry faculty member who wasn't really paid by the university, or he got grants and paid his salary the whole. He died a couple of years ago, but sadly. But he became very famous, and so one day I got my new Marshner, the Bible for plant nutrition. It was by my friend Marshner from Germany. And I looked in there, and I was looking through it, and there was one of Rich Guyette's publications.
Really?
And so I copied that picture. It was data right from his publication, a figure. And so I copied it, and I took it over to Rich, and he had no idea, And what it was is Rich, you know what he did for a living?
Yeah. Rings. Tree rings, yeah.
Tree rings. And so he went to he never would tell me where this was, but it was on the Current River somewhere, the oldest cedar, eastern red cedar. And he took cores of it. And then he took them down to the reactor center, and they did all the mineral elements at each core. And so what he found out that was way back in the 1700s, there was a lot of molybdenum in those cores. And then suddenly in the 1800s, it started going down. And it kept going down. And the sulfur when it was going down, the sulfur was going up
Hmm.
in the 1800s and then into the early 1900s, and then it sort of quit. So what was happening is Missourians were starting to use Missouri coal,
Mm-hmm.
which is high sulfur, soft coal, and there's more. We started burning coal and putting sulfur molecules in the air, and it was competing with molybdenum. So you could see that in these tree rings.
They tell quite the story, don't they?
Yeah. So, yeah, you say that, okay, rich guy that just counted tree rings. He did a lot more than that.
Oh yeah, yeah. Well, and now I think if we look at the sulfur, it's going down where we're getting a deficiency now.
Oh, yeah.
And that's, I was out in the cornfield this week, and I can see it everywhere.
Yeah.
Sulfur deficiency.
We need to start burning Missouri coal.
No.
I'm just kidding.
No, but it has opened up a lot of different research now.
Exactly. Yeah.
And-
I keep hearing this from a lot of people that it's time to start looking at sulfur again.
Yeah, it is. It is. It's a great quality, for our forage quality, our seed quality. So it's really neat. And I need to go back and look at some Sanborn Field soils and plants on molybdenum now.
Yeah.
I got the sulfur
Yeah.
profile. Every time you have a conversation, it leads to something else.
Oh,
doesn't it?
exactly.
Now, you also worked on magnesium. Now, what was that all about?
Oh, well, that's a very personal thing. Well, growing up on the farm, my dad was getting ready for me to go to college, so he thought the dairy may not have a future there because he was losing some helpers.
His help, yeah.
So he started breeding our Holsteins to Angus bulls. So we had Angus cows, and then he liked Hereford bulls for the Angus cows. So we had black baldies. So we had a kind of a business with black baldies there for a while. But anyway, our best cows, once or twice there, died of grass tetany. So I became kind of interested in grass tetany. It turns out that in the cow, it's a magnesium problem. They can't get the magnesium. So we started working on grass tetany. You started working on grass tetany. I think you did 25 different experiments.
That's right.
Before you figured out that, okay, if you're going to get magnesium in a plant, you need to first take care of the phosphorus.
Right.
So I thought that was a big breakthrough. But so we started studying grass tetany and the role of phosphorus in getting more magnesium in plants. And that seemed to work pretty well.
Yeah, and like you always said, you got to crawl before you walk. So we did those 25 experiments in the
Hydroponics.
Hydroponics, yeah.
Pure everything.
Yeah, no soil. We knew everything was in there.
Yeah.
And you'd run these experiments. And then once we found that, then we had to go to the greenhouse.
Yes.
And then to the field.
To the field, and especially the Southwest Center,
where it was a big prominent area.
Yeah,
for it.
Low phosphorus.
Mm-hmm. And then later on, you and Colin Bach and Ryan Locke did the cattle.
Yeah. And then we went up to Dave Davis and did 150 cows for 3 years and calves.
Mm-hmm.
The trouble with working up at the Forage Center up there is that the soil was a little low in phosphorus, but not as low as it is in Southwest Missouri. So, but anyway, we did 3 years with 150 cows, got nothing. But then the story gets more interesting because we terminated that experiment. But the next April, I got a call from Dave Davis saying, "Hey, cows are dying with grass tetany."
Oh no!
So we ran out, Christina, Missy, we all ran out, started sampling fescue. And it turns out typical, it's hard to study grass tetany in the field because you can't predict when you're going to get it. Well, that year we had this freeze, April freeze. It was terrible.
Yes.
And that's when we started having grass tetany, or people started having. So we ran out there. We'd had an early warm spring. The fescue leaves were that long. And when we went out to the field, the top half was brown and the bottom half was green. So we took samples, and we sampled the brown part and the green part. And it turns out the only thing that was different in the two was sodium. And it was much lower. It had bleached out of all those. When that bleached out, it was leaky, leaked all the sodium out. Well, so we published a paper on that, on grass tetany and sodium, because there was hints of that in England on ewes. They grow a lot of sheep over there. And also, I was sort of called quite a bit by a guy from Kentucky, a retired veterinarian who has been on my case for a long time about working on sodium. He's retired and doesn't do research, but he believes that sodium, in his experience, was a real key to grass tetany. So we got data for it.
All right. So it's sodium deficiency.
Sodium deficiency, yeah. So it's magnesium in the cow, but it's sodium in the plant, because sodium in the cow helps magnesium gets transported where it should be transported. So they're together, sodium and magnesium. You need magnesium, but you need sodium to get it in the cow where it's supposed to go.
Oh, wow. So, makes you wonder
now,
those Southwest soils, how they are in sodium.
Yeah. Because at that time, I don't think we even analyzed sodium.
No.
Or we might have had it, but didn't even pay attention to it.
Is sodium and potassium real close to the periodic table, aren't they?
Absolutely.
It's got to have something to do with potassium.
Yeah, it may.
Again, that's the kind of conversations that Dale and I used to have.
Yeah.
So,
but anyway,
that's great. That's fantastic. How that can continue to solve issues later on in those questions.
So I think there's some people that are critical of my suggestion about sodium, but I don't think they have data.
You know, I remember that a lot. People would be critical of something, or say, "Well, we've already done it," or "That doesn't work." You'd say, "Show me the data."
Yeah.
And they can't.
Yeah.
You know, that's one of the things that you taught us, is that whenever I give a talk, I either show my data and those people I trust.
Yeah.
You know, good data. Going back to our very original, the experiments were done correctly.
Yeah.
Because so many people will make statements based on opinion, not facts.
Right. Yeah.
You guys show me the data on that.
Hi, Dale mentioned grass tetany and how we had worked together on this. Grass tetany is a condition in the spring that lactating cattle often get. It usually affects the most heavy milk producing cows that you have, and it looks a lot like this, where they just get despondent. They get down, they can't get up, they're disorientated. And what they found was it's from a blood serum magnesium deficiency, and sometimes calcium. And over the years, we found out that high nitrogen rates can make it worse, high potassium rates can make it worse, but they didn't know how to overcome it. But the only thing they could really do was to have mineral blocks out there and hope that they would get enough just from licking the mineral blocks. Or you could keep them out of the lush grass, because that's when it happens often, when the grass is really greening up, really starting to grow. This is when you start getting some grass tetany. asked my father-in-law once if he ever had issues with it. He said, well, we just kept him out of that new green grass. Of course, you also are going to be losing some opportunities to take advantage of that grass. But what's happens is that you have an abnormally low magnesium level and also increase in potassium, because potassium is not limited. You get too much potassium compared to magnesium, and that's when you start getting some issues with her muscles as well as in her brain activity. And what we found, we started at the very basics, and we learned about what controlled magnesium uptake, and we found out it was phosphorus. We took this to the next steps, just looking at the forage itself, and found out, yes, if we could increase the phosphorus content of the plant in the early spring, then the magnesium levels would stay up. And finally, we did a study with livestock where we found that in the early spring, when the grass really started greening up, the magnesium levels did stay low, but when we had adequate phosphorus, and this is soil P phosphate in Missouri of 30 in the green compared to 6 in the controls, that we could get more magnesium in the actual blood of the animal. So this was a really important thing that we found, that we could help control grass tetany in our lactating animals and not have them die, because often if you didn't get to them quick enough, they would die, and they would give them a shot of calcium gluconate to get them back up and going. So grass tetany was one of those issues that is a metabolic issue in livestock that we help to overcome through our basic knowledge of magnesium uptake in the plant, and how farmers can help prevent having magnesium deficiency or grass tetany in their livestock, is to make sure you have your phosphate levels up to near optimum levels. That's in Missouri, that's about 40 on the optimum. Make sure you have it up there. And also, often phosphorus is tied up in the early spring, so if you put some phosphate fertilizer on in the spring, you'll not have that issue either. So just adding magnesium did not make a difference to the soil. You had to have higher phosphate and magnesium.
Now, you also were a big part of the graduate student seminar series.
Oh, yeah.
All the graduate students had to give a seminar.
Yeah.
And you and Larry Dara and Bob Kramer, that was,
yeah.
At least when I was going through, those were the three.
Yeah.
And you gave us a lot of suggestions.
Oh, exactly. So I think that's one of the, during my career here, that Missouri, and I have to also give Jerry Nelson a lot of credit, because he was one of the instigators in this. But later it was Larry Dara and others. But we ran a really good graduate student seminar series, and we spent the first meeting or two suggesting how seminars should be given and what you should do. And so then after every seminar we would have a critique right then.
Mm-hmm.
And it was very difficult for the person that had given the seminar to stand up there and take the critique at first, but then people got kind of used to it, and people tried to be pretty kind. I can imagine, I have nightmares about the first time I gave a seminar.
Yeah. It
was awful, because in my graduate program where I got my degree, they didn't teach us how to. They didn't even make suggestions. It was your turn to give the seminar, and you tried to give it.
Mm-hmm.
But I think here at Missouri, we did a good job of telling people what to do and how to do it, and then with the critique. And as a consequence of that, when later on the Agronomy Society started having graduate student presentations and awards at the national meetings. So it was a competitive, like we were very competitive with Purdue.
Yeah.
You know why?
Yeah.
Jeff Olenek was there. But anyway, so my students won a lot of first prizes.
Oh, yes.
and other students from Missouri did that too. You know?
And then those students went out and they taught seminar the same way.
Yeah, exactly.
Because you used to be able to go to those national meetings and pick out either every Missouri graduate.
Yeah, yeah.
Because they were so good, like you said.
Yeah, yeah.
But then it got to be, well, Purdue was doing it, Iowa State because they learned from you all.
Yeah, yeah.
But that's a legacy, and it really has upped the type of presentation that you hear.
Right, exactly.
And that's so important.
It's very important.
I remember you told us, if you got you gave an example that one of your friends had to practice it 24 times.
Oh yeah. And this friend got his degree at Harvard.
Oh, really?
Yes.
Oh, wow.
So he was a pretty smart guy, but boy, he practiced like crazy.
Mm-hmm. And at first you have to, but then you get good at it.
Yeah, yeah.
And
well, and then some people like this friend, I think he had a little hard. He had to work hard to do it well, and he did. You know, it didn't come very. Some people it comes kind of naturally to get up and give a good talk. They know how to talk, and some people are shy and laid back.
And we've seen people come out of that shyness. They can give it.
Yeah. Well, yeah. One of I could name her name, but she's been a professor at Utah State for many years. She was very laid back and quiet and shy, and she became excellent. Right.
Is both teaching and giving talks, and yes. Well, now what other type of what other research we've covered quite a bit. Now, toward the end of your career, you started working with some roots, more in root biology too, didn't you?
Oh yeah. Well, I think and we had shared a huge lab area with Bob Sharp, and he was a root guy. But roots are difficult to study.
Right, they're underground.
You have to dig them out, and it's not so easy. It's pretty dirty. You have to wash them, and it's kind of hard.
A lot of messy stuff and Elizabeth stuff.
They
did a lot of work on.
We did a lot of work on that, and
trying to get the mechanisms down,
and calcium and liming and pH and all that is really important in roots.
And even though we already knew the basics of that, how that all fit together was so important on the plant side.
Yeah. And there's deeply rooted plants and shallow rooted plants, and when they grow together, you wonder about what leaks out of the deeply rooted plants and the shallow rooted plants can take up. And
it's amazing. They're telling us now, though, even the plant itself will feed the soil microbes, trying to get certain soil microorganisms there. Yeah. So there's, what we're learning is just tremendous.
Oh, yeah. There's one thing that's always been, it keeps popping up in all the research that I did, is the fact that the simple organic acids like malic acid are so important and feeding the microorganisms, and also as a companion once they get in the plant for potassium movement and stuff like that. Yeah, they're often overlooked. I think organic acids are really
so important.
Citrate and malate are the big ones.
So, what do you want to conclude with? What on research or students that good advice to our future graduate students?
Yeah, well, I think one thing that I've always thought was really important is reading, and I'm trying to keep that. Tell my grandchildren about that. Reading is really important to keep up with the literature, and also for getting ideas. I think, and knowing what's going on around the world and what's in science, and I think that's really important. And then you got to stay interested, really. And the hardest thing now, I think, is getting funds. It's been hard ever since I can remember.
Right.
The percentage of winning in a grant is, below 5, a lot of 5%
Yes.
in grants. And writing grants is really difficult, I think.
Yes.
Good ones.
But if they keep up on the literature and try to put the mechanisms together, they can go a long ways.
Yeah. And then as we've talked about already, is drawing a model somehow.
Yes.
And it'll help you with your grant writing and also with designing your experiments.
It's kind of like building a house. You gotta get the plans first.
Oh yeah, exactly.
How everything
Exactly.
works together.
Yeah, exactly.
Dale, we could go on and talk about lots of things. I probably dominate too much,
No,
but you've had a fantastic career, and you're still working on things.
yeah.
like putting a lot of this together. That what we're missing still out there. And
Yeah, I'm thinking about aluminum and boron right now, and the fact that they may not be boron may not. It's an essential element, but there may be more to it than that outside the plant taking place.
We're finding. I'm still doing some experiments, and we can find if we put boron on corn, we find it in the grain. It increases, so the plant is utilizing it. So there's a lot going on.
There's something going on, and
There is
still a lot to know.
not only the structure, but other parts of it.
Yeah,
The sugar transport, and
Yeah.
so important.
Yeah.
And the interaction between them, that's what's fun.
Oh yeah.
So, all right. Well, this is it for Tim's Take. We've been with Dr. Dale Blevins, had a wonderful discussion. And as I said, we could have talked hours more, but we might have wore ourselves out if we done that. All right. Well, we'll see you next time. And thanks again for Dale for coming out for Tim's Take. Bye.
Wow. Well, that was fun.